Hey, everybody, and thanks for being on today on this long weekend Friday. I just want to talk a little bit about Dr. Sysoeva's very interesting background. She was actually trained as a material scientist where she received a bachelor's and master's that were earned at Moscow State University in Russia. She changed to biological research after this to study how bacteria adapt to their changing environments and interact within microbial niches. During her PhD studies at the Pennsylvania State University and postdoctoral training at Harvard and Duke, she analyzed bacterial transcription regulation, transport mechanisms that are crucial for bacterial adaptation. This included studying protein secretion and DNA transfer processes using bacterial genetics, structural biology, biochem, and bioinformatics. DNA transfer was particularly noted to be critical to the spread of antibiotic resistance among bacterial pathogen. And she transitioned her studies to the characterization of clinical antibiotic resistant pathogens and their interactions with commensal microbiome communities. Tanya is currently an assistant professor at the University of Alabama at Huntsville, Department of Biological Sciences, kind of my old stomping grounds, where her research group focuses on the problem of multi-drug resistant urinary tract infections, something that's really of high interest. They approach this problem by studying natural processes such as plasmid exclusion that prevent spread of resistance, and by characterizing human urinary microbiome that may hold an answer as to why some patients are more susceptible to UTIs than others. Her goal is to develop novel strategies to prevent and treat antibiotic-resistant UTIs via drug or actual microbiome interventions. So thank you for being here today, Dr. Sysoeva, to discuss with us urinary microbiome resistance, potential defense against UTI. Thank you so much.
Thank you so very much. I'm very honored, humbled, and excited to be presenting. But before we jump in, I do want to kind of get a really brief but introduction. What kind of audience we have? How many of you took microbiology classes? So just want to have, okay. So you may have some flashbacks, right? And I will try not to fall completely into my lecturing mode because I'm used to teach microbiology and sometimes I'm kind of going to the lecturing mode. So let's try to be more interactive. And if there is any, like my request, please try to do the raising hands or some other things, right? And if there is a disconnect, please somebody call me because I've been in these situations before and it's not nice. But with that, again, in this room or Zoom, it's really probably not needed to introduce that, but the sheer number and importance of urinary tract infections is overpowering. And usually it is regarded as a very benign condition. It's usually self-resolving. And while there is millions and probably more millions than reported worldwide and several millions as estimated in the United States annually, it's still considered to be very treatable by usual course of antibiotics infection. But the problem is that bugs are not sleeping, they're always evolving and they are gaining resistance. And again, it's not something new to you all being medical professionals that we do have huge increase in multi-drug resistant organisms, including uropathogens in particular, because we have high increase of our risks to get UTIs if we are in hospitals or if we have immunocompromised hosts. And because the sheer number of antibiotic prescriptions which are given for the usual UTIs, even though many cases they won't be considering to be life-threatening or go into life-threatening conditions. But the antibiotics are the only strategies. We don't have any other treatments. And though there are new developments, right? There are new trials and some very successful trials about vaccines and other treatments, it's really not yet out there. So combination of the sheer number and the rise of drug resistance really brings this up front. And some people very eloquently, hopefully with my accent even more eloquently, bring it to attention that AMR, antimicrobial resistance in UTI, is an unignorable health problem anymore. So, and it's truly the case because recent reviews putting that overall the main causative agents of urinary tract infections, if they are multi-drug resistant, can cause over 800,000 or over 1 million deaths annually. And that comes as a basically cold shower saying like, yeah, it's rising, it's a problem, but it's already here. And many people refer to this as a silent pandemic. So that's how I brought to this research, because I'm studying how drug resistance spread. And I was kind of dragged from what Dr. Richter called from chemistry to structural biology to molecular, and then basically almost at the organismal level. But I'll try to connect it more to the clinical aspects of this, because I think we are close to basically trying to find some different solutions. I'm being too hopeful. But that being said, some activity, exercise, prayer weekend. We know that urinary tract infections are caused by Enterobacterium. So the paradigm in the film, they're all coming from the gastrointestinal tract, and they are migrating and causing this ascending infection in the bladder. And the main uropathogen, those bacteria which are causing the infections, are known suspect. Uropathogenic E. coli, or UPEC. You'll hear about this a lot today. Klebsiella pneumonia, Enterococcus faecalis. And if I may ask, we are all familiar with the gut microbiome, and there is lots of microorganisms expected to be in gut environment, skin environment, vaginal environment. But who knows about the old paradigm that urine is sterile? And let's not mention about how to treat wounds. Who knows, probably read some of the Dr. Ritter's work, that urine is not sterile. Can you raise your hand just so you have some feedback as I go? There is a special emoticon button now, right? So you can basically give me some sign. So if you're familiar with the suspect that urinary tract is not sterile, thank you, Holly. Anybody else? So when you were taking microbiology, I'm teaching still on textbook.
So again, in the name of just spitballing ideas, it takes me time to digest this stuff. But I'm still, I have an arm about the numbers.
Okay, but anybody on Zoom, there is a button down there. If you mouse on the bottom. So basically, do you know that the urinary tract is not sterile? Okay, we got 2 responses. Okay, getting there. Okay, getting there. Thank you very much. So, and that basically oh, yes. Okay, I know it's not standard, and hopefully we'll get more questions this way and discussion. But that came by surprise about 6 years ago for me, because I was told by these textbooks I'm using, and I'm trying to contradict textbook in my classes, because we learned that it's not true anymore. So that paradigm is changing, and basically these niches in particular are populated by microbes. Majority of microbes are bacterial cells, right? And for last 12 years, about 12 years, if I'm putting it correctly, we're trying to explore who lives in the bladder, urethra, and all other parts of urinary tract, and basically try to characterize what are they doing there. And we have some major discoveries were made by the groups, including UAB, showing that, for example, in women, this microbiome is populated by lactobacilli, which is a several genera of lactic acid forming bacteria, which is similar to the vaginal environment in females. And we were lucky to collaborate. And here's a presented study from Dr. Wan and Dr. Siddiq from Duke, showing the example how the lactobacilli dominate this environment. And I will bring you more about this bacteria information. But here, I want to draw your attention just as an example that the 2 cohorts of patients were in this case, postmenopausal women, which either have healthy urinary tract or were diagnosed with recurrent UTIs in the past. And the picture here is summarizing the abundance of different bacterial species in their urine, which was obtained by catheterization, which is a golden standard now in urobiome field, which basically only highlights the different species of lactobacilli. So the colored bars on these plots for no recurrent UTI patients showing who lives from lactobacilli genera in the bladders of these patients. And what you can see that majority of patients, which are different columns in this relative abundance plot for microbiome study, is showing that majority of women would have lactobacilli of different kind in their catheterized urine. And in those female, it's a really big chunk of different, comparison with other bacteria in their bladders. And what also I want to draw this attention that they also different combinations. And majority of women, so different bars, would only have 1 or 2 different species of lactobacilli. And also draw attention that it's very diverse. So basically, patients would have differences, but we can distinct that, for example, more green presented the recurrent UTI plot on the right versus no recurrent UTI plot, which presented mostly by these bluish bars. So that was the first in initial studies, and now more studies available showing there might be some differences in patients who are having recurrent UTIs versus those who are not prone to have chronic or repeated urinary tract infections, that they have different composition of urinary microbiome, and in particular lactobacilli. That was probably a very steep introduction for the microbiome. But we also know that there are different conditions, benign conditions and oncogenic conditions in urinary tract, which are affecting the composition of urinary microbiome, including that there are some leads now. There are different lines of evidence that saying that predisposition to urinary tract infection and their recurrence may be dependent on the composition of urinary microbiome. And it's not clear whether that's a causative, correlative, or it's a post effect of recurrent UTIs. But we do know that now that we have all these different microbiomes, and therefore when uropathogen is causing the infection, so it will relocate from one niche to another space in the body, it goes and have to interact with different microbes. So basically, nothing is in a vacuum. There is always some interactions, and it's critical to know that uropathogen is populating niche, which is not sterile, which has somebody who is resident there. And therefore, there is a next question possible. We know from other microbiological niches that if somebody is already occupying the ecological niche, then they probably don't really want to lose their food and access to resources with any newcomers, for example, pathogens. And therefore, the question for microbiologists like me, and being like really paranoid about different bacteria and microbes, there comes the idea that if there is somebody in the bladder, can they actually resist urinary tract infections? And we are trying to figure out the answer to this question. We have some leads, which I will be presenting as part of 2 different sub-projects going on in the group. So the first, we were very lucky. When I was a QRA scholar at Duke University, I collaborated with Dr. Rand, Dr. Siddiqui, Dr. Kelly, and one particular study, the result of which I kind of showed a little bit in the front, showing that there might be differences between recurrent UTI and healthy patients, postmenopausal women. We were lucky because we not only contributed to analyzing this, but we also were able to collect the whole repository of urinary bacteria. Small repository, only 74 different isolates, but they contain so-called enhanced quantitative urine culture technique to isolate microbes from this catheterized urine. And we had 3 cohorts of no UTI, recurrent UTI, and recurrent UTI patients on prophylactic antibiotics. So we had representative isolates from asymptomatic individuals who are not currently experiencing the infection. And we were very calmed and felt justified to obtain that 27 out of the 74 isolates were lactobacilli. So there was those bacteria which seems to be dominant, and they were coming from 7 different species. So we had a good diversity of these bacteria presenting from this species, L. crispatus, L. gasseri, Jensenii, iners. Those are the ones which are most abundant, which Dr. Richter and many others show that that's basically the core compositional lactobacilli which are met in majority of women. So, and therefore, as a microbiologist liking to do everything in vitro and do some mechanistic study, we're happy because now we have this representatives of urinary microbiome, and we can ask more detailed and mechanistical question why there are some patients which potentially have no UTIs and some have recurrency issues. And here, maybe too late to the game, but I want to introduce lactobacilli, which is generally known as a generally regarded as safe or grass organism, and also as a good bugs. And if you have lunch or just had lunch or maybe had some of these foods for breakfast or taking some probiotics presented here, you're probably already well familiar on daily basis with these good bugs because humanity used, and I'm not overstating, for millennia, these bugs to do the food fermentation, preparation, preservation, and they are now used in many countries for the gut health or food additive for the livestock, and now even used for vaginal health, and not surprisingly, but surprisingly, for example, for lung health, right? So basically, having these good bugs around seems to be affecting many other things in our organisms, apart from just preventing our food from spoiling. So, but why, microbiologically speaking, they're good bugs? Because they're really great competitors, meaning they're killers. In bacterial world, they produce lots of noxious stimuli. They produce lactic acid, which is acidifying or lowering pH of the environment and kills majority of bacteria around because they can withstand it as bacteria can, so they overcompete those ones. They produce hydrogen peroxide, they produce different protein and peptide toxins and other toxins, bacteriocins and others. They kill on the spot other bacteria by different mechanisms. And so combine all of this together, they're good bugs because they're good killers of competitors, including pathogens. And therefore, from this perspective, knowing this characteristics of this bacterial genera, we can basically restate our question of whether how bladder microbiome interact with urinary tract infection and pathogen by asking, okay, we have the representation from urinary bacilli. Do they look differently from other lactobacilli, for example, from our food or vaginal environment or gut environment? Do they interact directly with the pathogen so they can actually directly interfere or not with infection? And then whether they interact among themselves and within the niche to establish different composition of microbiome. So going further to trying to restate it, we literally ask some of this question very directly, meaning that if you want to ask whether representative lactobacilli from human urinary tract can interact with the pathogen, we can set up binary assays at this stage. So simplifying and going reductionist approach. And we can, for example, use the classic microbiology assay. Probably heard about that in antibiotic testing, other testing, when we can just have something which we think may be inhibiting growth of the pathogen, and then interacted with the pathogen which is distributed on the lawn of the plate. You're probably all familiar, but I'll still go with the schematics. This is the Petri dish on which we loaded a target bacterium, for example, uropathogens, and there's great brownish color beige, this signifying confluent growth. So we have a lawn of bacteria, and they're happily growing unless we put in either holes or wells, whatever you call it, with some chemical or other bacteria, for example, antibiotic. And then if we increase the amount of antibiotics or other inhibiting agent, we can see zones of clearance, which we can measure. And then if we have more inhibition of growth, we'll have more clearance zones. So it's a very classical assay. It's used in clinical microbiology to see the drug resistance and so forth. But in our assay, we can simplify our binary interaction of urinary microbiome bug and the uropathogen and ask the question, do they interact and whether one of them can kill another, right? In this case, we'll just have our target of uropathogen in lactobacilli culture in the wells or loaded on the surface of the agar. And that will be the only schematics I show because I will be showing process data when we literally quantify the inhibition zones if we observe inhibition, except this plate. When we tested just domesticated, non-pathogenic E. coli, G1655, our control, and we took the first two lactobacilli, which we had isolated during the repository collection, and it's happened to be Delbrueckii and Gasseri of these numbers from the patients. And what we observed that there is inhibition. That was the first undergraduate observation that, oh, it actually works. So we took this assay, and while we now switch into this, the result of first half of the talk would be all regarded to the inhibition assay. So now you're familiar with that. So what we did, we took classical uropathogenic models. So we have three major pathogens, UPAC, E. coli, Klebsiella, and E. faecalis. And we took several strains of which, if we could get our hands on this and tested, again, these two, Gasseri and Delbrueckii, lactobacilli from human, two different patients. And we were very excited because for majority of these uropathogens, we saw inhibition, which we kind of expected, but it was still very exciting to see, except gram-positive E. faecalis was inhibited significantly less, which is also expected because gram-positive organisms often are more resistant in general. And then we tested different conditions. For example, here, different color represent different timing of how we prepare lactobacilli, mimicking that potentially how they grow different in different areas. And then we make an observation, which got us really excited because the Gasseri, which seemed to be a stronger inhibitor of these model strains of uropathogens, came from the UTI healthy cohort, while Delbrueckii came from the recurrent UTI cohort. And we come up with very simplified hypothesis. We thought we can test that maybe the inhibition strengths of the uropathogen by the urinary lactobacilli would correlate with the health status of the patient. That would be so cool. Then we would be able to easily predict whether each particular individual's predisposed to recurrence of UTI because maybe they have a bad inhibitor in their bladders. So we took all of our culturable lactobacilli from the bladders. We couldn't get all 27 because we wanted to culture and grow them under similar conditions in MRS media. And we tested all of them here against E. coli CFT73, the classic UPEC strain, which is studied by many groups as a model. And here we grouped all of different lactobacilli species in a groups of different strains, like each species, several strains. For some of the species, like Johnson, we only have one strain, so it's by itself. And we had a positive control of yet another lactic acid forming bacteria, but it's not lactobacilli, which is in this case Strep aeruginosus, which is very broadly presented in urinary tracts as well. And what we observed, the first obvious observation that many of them are inhibiting uropathogen UPEC. And there is a variability. Even within the species, even if you have several representative strains are differing. So it seems to be very nice. So majority of urinary commensals can be inhibiting uropathogens. We expanded the screen and we used top 52 for Klebsiella and OG1RF strain for E. faecalis model. And we observed that again, this pattern that majority of different species of lactobacilli inhibiting classical uropathogens stands. It was very cool. So now we can test our hypothesis by asking where these different isolates come from. Do they come from the recurrent UTI patient cohort, or do we have somebody coming from the healthy patient cohort? If we label them differently, we can overlay, and you don't have to count. You will just say that, oh, they're very distributed randomly, it seems. And it's true because when we do other calculations, for example, we take different cohort isolates, and we even group them for against different pathogen. There is not much correlance. There is no statistically significant difference for the strength of inhibition against different pathogen if the lactobacilli isolate came from the healthy or recurrent UTI patient, which kind of showed that our initial hypothesis was not correct because there is no correlation. There might be trend, but because we only have a few number of species or strains for each species, we can't make a strong conclusion. So, and we can only guess at the moment why we don't see correlation. So possibly those binary interactions are not providing the full picture. But we do test the differences in what mechanism of inhibition these different strains of lactobacilli have. And here's a snapshot showing that just two those initial strains, Lagasse and Delbrueckii, coming from the healthy and recurrent UTI patient, showing the test for what kind of components of the lactobacilli actually inhibiting the cells, just to show that in case of gastric culture, we can just take the clear cells and they would be inhibited by some cell-dependent mechanism. We're still struggling to understand what exact mechanism is. And if you take the cell-free filtrate, so whatever the cells excreted outside, including lactic acid and low pH, we can show that it's not bacteriocins because if we just adjust pH, we're losing all inhibition. I do apologize. And similar happens to Delbrueckii, but in Delbrueckii, the cell-dependent mechanism seems to be not present. And if we consider that the urine is very highly variable environment with change in pH, it might make sense because some of the strong inhibitors have complex inhibition mechanisms. So we have different ways how lactobacilli can kill pathogens. And it becomes interesting because we also can try to move to more physiological state, for example, to liquid culture versus growing it on a plate, right? There is no plate in the bladder. And we can see if we co-culture uropathogen with lactobacilli, so kind of getting closer to mimicking what happens in the real bladders, we see that we can test different ratios. If you have more lactobacilli, fewer lactobacilli in your bladder, then your uropathogen will be killed with different dynamics, right? So you can see here that we took no lactobacilli and 1 to 1 to 1, 1 to 5, 1 to 10, and 100 different lactobacilli, then we can kill either all or majority of E. coli in the liquid medium for using like lactobacilli gastrate. But if you're using different species and different strain of lactobacilli, the killing still happens, but it's now significantly weaker, right? So we need to test more strains, and we need to understand which of them use simple pH-dependent mechanism of inhibition or otherwise to actually expand. And maybe our hypothesis is not that bad. We just don't have a sensitive enough method to understand that might be difference between different lactobacilli isolate coming from healthy or recurrent UTI patient. So the summary one for that, and I only have 15 minutes for the short introduction. Second one is that we find that urinary commensals in humans actually capable of inhibiting growth of major uropathogens. And this inhibition works better for gram-negatives, but what I didn't show that also works good for clinical isolates, for multi-drug resistant isolates, because they're not relying on antibiotics, on different kind of interactions. And there is a huge variability in species and strain levels. Therefore, maybe one particular species is not fitting them all. And that might be very important to characterize and find the best, phenotypically best lactobacilli. And interestingly, some of the lactobacilli seem to be having a combination effect of different mechanisms. And we want to go and study it deeper to understand what would be the best potential protector of urinary bladder niche. So with that, we are thinking what's next. And I'll present two small directions. One in particular, I'm very excited to present to your group, because we're trying to understand that urinary microbiome is complex. We know that it's not only a couple different species, it's a combination of bacterial, some fungal, and some viral species. So we cannot just use two species and hope that it will recapitulate the whole interaction, even though hoping we'll find some driving interaction. And we did use the bugs isolated from the postmenopausal bladders, and we do know that the postmenopausal females are more predisposed to recurrence of UTI, and the immune system works differently, and the microbiome is changing. Therefore, we're hoping that we'll be able to add the complexity, and they basically keep answering the question, do we have resistance and how we can use it for the treatment strategies. But in addition, because we're thinking and hoping that we will at some point figure out what kind of clinical trials and what candidates to use to try to create probiotic or the treatment microorganism which would modulate the urinary microbiome, the whole books are written about trying to ask what actually good probiotic would constitute. And here I'm presenting just two pictures, and I'm not paid by them, just took from the internet, showing Lactobacillus rhamnosus, which is actually present in quite a few bladders of people analyzed. And usually the conclusion that that organism should be able to colonize the niche. So we don't have to constantly retake and repopulate the niche. It should actually survive the delivery, for example, by oral probiotics, of course, very important. And it should also compete with the present microbiome in the niche. So we're testing this because we know, and remember I pointed out that all these bars contain only few and usually just one species of lactobacilli. And we also know that usually when lactobacilli and any other species, they compete, they mostly compete with close by species because they are having very similar metabolism, therefore they compete with the same kind of resources. So, and it's not surprising that if we will test and see what kind of lactobacilli would overcompete. Why it's important? Because if, for example, the patients in general populated by species such as, let's say, Iners or Crispatus, maybe getting some Lactobacilli rhamnosus probiotic would not be a good idea because the resident microbe would be out competing. Therefore, we wouldn't have a colonization with probiotics and the protective effect, which potentially can be provided. Therefore, we did the pilot studies now. We screen our lacta-alacta interactions, so mena-mena interaction with using similar assay at the moment. So we took the best inhibitor which we had in our collection, which happened to be Lactobacillus rhamnosus, but these ones are coming not from the pill, not from the pharmacy, they're coming from the patients. And we competed them against other lactobacilli coming from the urinary bladders. Here, Delbrueckii, Gasseri, Johnsoni animalis. And you can see that among this, rhamnosus can actually very well inhibit, for example, Delbrueckii, but not that much other species. We have a luxury of having several different strains of rhamnosus we tested, and it seems to be, while it's variable, we still have a species-dependent inhibition, which we're still figuring out the mechanism of this, but it's promising. It's also kind of interesting to observe because we didn't select Rhamnosus because it's the most sold probiotic probably, but just because it was abundant and we had quite a few of them in our collection. So we had some variety to test. And so we do know that now that it's not pH-dependent inhibition, and it's more close contact or cell-dependent inhibition. So we're going to move with that forward, trying to address, can we propose a probiotic candidate at this stage already, combining our data. But we're also trying to figure out what is changing in the urinary bladder development. So we're trying to figure out the best protocols. So we published and trying to figure out what kind of DNA extraction process to use, what kind of urine collection or biomass isolation from the urine methods to use. But in particular, we're very interested in figuring out the development of this particular microbial niche with age. And we know with the premenopausal or postmenopausal women, we have quite a lot of data, but it happened to be that children microbiome in urinary tract is almost uncharacterized. We started our studies 2018. There was no publication on that, but of course, with slowdown of COVID and everything and difficulties of study children population, we only now have the results, which I'm going to show it to you. And now we have about 7 different publications on urinary microbiome of children. So luckily, we're getting some new information to compare. And we're trying to do this investigation at the species level because of the first part of the talk, when I'm showing that we cannot just use usual 16S sequencing, which usually does not provide species level resolution, and try to make conclusion about the protective effect of the urinary bladder microbiome. Because we just showed that even going with the same species and different strains of that species, we may see quite a few differences. And therefore, it's very important that we want to take fine resolution of who is living in the bladder microbiome. But again, you being specialist, it's not for you, for me to describe to you the difficulty of studies, because the ability to collect voided urine or clean catch urine in children population is almost impossible at the very beginning of the child development. We look in this part of the spectrum. And later on, it's very hard, at any point, almost hard to actually collect healthy individuals or background individual urinary catheterization sample for obvious risks associated with the study. Therefore, majority of the study, including ours, is including either convenient samples or specific populations to collect the catheterized urine. So in our case, we were using VUR patients who are medically indicated to undergo catheterization. But we want to basically start at the very root of the problem, how the microbiome is forming, in order to see which individual later in life would be susceptible or not susceptible for recurrent UTIs, right? And with the hypothesis underlying that the composition of urinary bladder would be contributing to that susceptibility. So in our convenient sampling, which we started 2018, I cannot believe it, just before I moved to the new position here in Alabama, we connected with Duke Hospital, a neurologist, in particular, Dr. Kelly, and we collected samples during the VUR testing. So whenever we get in cystic ureterogram to see if there is a reflux, and we were able to, for 3 years, to collect only pilot study for 52 children. And then we received funding to sequence it with the species level resolution. And now we're running 2 additional studies, which would be answering couple other questions we're hoping. Because we're hoping that we can switch to the void urine analysis and basically test a wider range of ages with children population. And we want to try to get in the temporal resolution of the studies, which is almost impossible to do because of the pediatric population and the complex samples, because they're very low biomass. So we're testing different stability and storage samples by doing the time series collection of very small population from both sexes. And basically we have almost a week of collection, which is stored in home freezers, or which will be stored on site, and people from the study need to pick up them every day, every week, and then every month. And we're hoping to basically cover all our bases to design a larger study to collect urine from these patients who either already at this young ages have recurrent UTI or those who are healthy. And that would help us in both cases to establish the baseline with what pediatric urobiome looks like, as well as what is setting up those poor children who are having recurrency with the returning UTIs. And I'll present the results from the first study because second one is we're just completing the collection. It seems to be going significantly faster than or faster than with the VUR, which seem to be convenience sampling. But we have very promising results. So hopefully we can get at the answer in the question whether urinary microbiome differ in children who are predisposed with recurrent UTI is also healthy. So with that, the first study, which we used the VUR patients to collect the urine, and we try to improve the method of collection because the complexity of this long belief not existing urinary microbiome coming from that it's very low biomass microbiome. That means that there's not many cells are present in any given volume of urine, and in particular pediatric is a small volume of urine can be collected. So we collected on average 13 milliliters from our patients. And we had patients with a broad range from 3 to 12 years of age. And we stole the method which is used in water microbiome and other very, very dilute microbiome samples. So we used filtering. So basically, we used filtering in order to get the liquid urine out and collect all biomass on a filter, from which then we can purify the total DNA and submit it for different types of sequencing. So this step is unique. And so we had the first test run, which I'm now publishing, to show that actually works nicely for urine, especially for the low biomass pediatric urines, which has fewer bacterial load than in adult population. And here we present result of very unique type of sequencing, which is just ramping up in the last 4 years, so-called loop sequencing. And I'm not going to bore you with details of that, but the idea that is method which requires to avoid contamination at many stages, and it allows you to sequence the whole 16S ribosomal DNA gene. And that is important because that contains, if we sequence the whole 16S gene, then we can contain more information, which would allow us for the species level resolution. And that's different from traditional 16S sequencing, which only taking small portions, so-called variable regions from the 16S RNA gene in order to identify who is there in the microbiomes. So here, just example of 3 different patient samples, which we sequenced in triplicates, quite inexpensive treat, but showing that if we sequence it in triplicate, we get in the resolutions which are very, very similar, which was technically, it was a good technical replicates. And it was very reassuring as well with controls that we can sequence the lowest volume, the lowest possible urinary biomass samples, and we can read them, not presented here, but later as a species level resolution. So it seems to be the method of filtering combining with loop sequencing, so-called synthetic long read sequencing, is working, which was very exciting for us. Because then we can take and see whether the boys and girls have different community. And the first statement, which now shown by several different papers while we were working on this, that there is a urinary microbiome in children. And I think the lowest age was 2 weeks in one study reported and 3 months in our study. And it contains several different OTUs or different species. So it's a complex microbiome. And it looks to be, if we plot it in different age groups, then you can see that this alpha diversity value goes up. So it is not a very clear correlation, and that correlation is mainly driven by the male population. So you can see that pink dot from the girls, they are all distributed at certain level, and then this greenish cyan dots for the boys, they're going up with age. We don't have many samples to do statistical analysis because we only have cohorts of 52 people and we rare factored to analyze only 33. We don't, unfortunately, have a cohort of boys under enough to analyze below 1 year of age, which would be very exciting to learn because this is the most susceptible boys as they get in the first year of life. And then the more risk for UTI, of course, coming from the female population. But it's interesting to see that we can see correlation that boys are getting colonized with the age, and it seems to be girls are starting with a higher diversity of microbiome at the very beginning, very young age. We also can compare diversity of this niche for the boys and girls, and we can definitely see that female urobiome at very early age already more diverse than the male microbiome, right? There's two different metrics for diversity, and there's Simpsons and Shannon showing that female diversity is higher than the boys' urobiome diversity, which is some sort of relevant for what we see from the adult studies. And we also can now narrow down to the species information. And I'm checking the time, I'm sorry. And we do find there are some different, even in our small pilot subset, there are different species, for example, Shaalea, Privotella, and Anacoccus species, which seems to be dominating in one sex. So in this case, in girls, and they're not present in boys. So we can now see at different species level who is populating which sex, and therefore try to correlate it with different clinical perspectives. And here's just an example of like showing all of the different compositions as a probably usual for your representation of relative abundance when we have stacked bar plots of different genera presented in the boys and girl cohorts, right? And one thing, while there are some statistically significant observations, some of which I showed on the previous slide, you can also see that there is a lot of diversity. And that's similar to adult population, that bladder microbiome is very diverse from person to person. And therefore, we certainly want to expand this study and do several site collection, hopefully in different states as well, and try to basically expand and to figure out what's the basic ratio of that. I'm not showing here, but there were several different other conclusions. For example, we do see, but my timer is off. So we do see that presence of, reoccurrence of the UTI, even in pediatric population, seems to lead to changes in microbiome. And those patients who have 2, 3, or more repeated UTI at very young age, they seem to lose the diversity of urinary microbiome. And it's an interesting correlation with other dysbiosis observed in other different niches. We also see that using antibiotics doesn't prevent colonization of urinary tract, and it seems to be not driving factor at certain age, at certain time after antibiotic course, which was very interesting to find out. And so the summary for this part of intercompetition of lactobacilli in trying to study development of urinary microbiome in children is that we do have a strong competition happening within the bladder niche among the commensals, such as lactobacilli, and their complicated interactions, which would be very important for development of treatment and probiotic strategies. And we do know now better methods to how to characterize this low biomass microbiome, even going to the very young population of patients. And we definitely want to proceed with more of that. And that urinary tract is getting colonized very early in life, and it develops with age. So with that, take home message that human urinary tract is definitely home to a complex microbiome, majority of bacteria, and that has ability, at least chance that it will be interacting with the infection development, so with incoming uropathogen, and that we need to characterize more detail and thoroughly the native urinary microbiome development and its property, so we can actually figure out what kind of probiotics or what kind of urobiome modulation mechanism we can propose. And with that, I would like to thank several undergraduates who partake in this first study and the published part of that, Lydia, James, and 2 high school students, Vaishal and Madam, and another undergraduate, Kaylee. And of course, our collaboration, Naz and Marilyn at Duke, and Lisa and Erin now in Oregon Health and Science University. And of course, our funding sources, on all of you for attention. Thank you.
Thank you so much, Tanya. That was really cool. And we have some time for questions.
That was the whole.
Throw it open. I'll start off. We have done some work in this area. looking at risk factors associated with recurrent UTIs. And, you know, I think we all kind of know in our minds um, certain types of things inherent to the patient that may predispose to infection. You propose in a way a new paradigm for the treatment of recurrent UTIs, in that I think you propose to kind of increase the proportion of good bacteria um, compared to the bad bacteria, rather than kind of treating bad bacteria. Can you comment further on this? You showed your probiotic. What are further thoughts there that you have?
Oh, I have too many thoughts on that, but I'll share some of that because this is amazing question, and we keep thinking about this. And we have now 10 students, 9 of them are girls, and who are really actively thinking about how we can use this new information, because we're thinking that maybe not the quality or the quantity, but the quality would be important. So we're thinking if we can colonize a bladder with a particularly good inhibitor, and if we can make that inhibitor stick, that will be our ideal idea of a probiotic, right? So to colonize somebody who will be very well adapted to the niche and basically kill everybody on the spot when they're trying to ascend in urethra and colonize the bladder. That would be the bottleneck which those probiotics should be solving. But at the very least, we want to move actually to answer our hypothesis, correct or not, whether the presence of a quote good commensal is helping or not helping, or is it predictive of a individual being predisposed to the UTI? And that kind of goes to like, we probably should be starting with diagnostic tools first, right? And see whether there is a correlative between, can we detect rhamnosus being present in a particular patient and say, oh, most likely you're not predisposed to recurrent UTI, right?
Mm-hmm.
And the second part, I would think it's how I take thinking about your question, is whether the present good bacteria or probiotic we put in the patient would be there to stay, because it looks like they're good competitors, and there might be already good competitors in there, because that's why they're resident microbes, because they're not allowing anybody to come in. We want to make sure that we are replacing or modulating the correct way. So we're not just putting somebody on top, but we displacing a bad microbe, which might be there. For example, we like the Delbruca, which we see there is inhibition, but not much, mostly pH-dependent inhibition, though the simplified prediction that it probably wouldn't be good if patients colonized with this organism.
Any questions? Please. I'm sure somebody has some. What are your thoughts on the role of estrogen in modulating good versus bad bacteria, both in?
First of all, there's definitely, we don't want to disregard the host completely, and we certainly don't want to disregard the adjacent microbiome niches. So I want to, if I may, two-part answer to that as well, because there's definitely a lot of information about how, let's say, using estrogen creams and other treatments alter the composition of vaginal microbiome, and now we know urinary tract microbiome, and increase the lactobacilli populations, right? So it would be kind of trivial to suggest that it's, of course, protective, and clinically that's what's seen, right, by many studies. And we don't know how it affects yet at species level. So we have a lot of field to cover to basically do the lab studies, right? Mm-hmm. So how each particular species or different commensal would behave in the presence of high hormonal levels. And so that's the hope that we'll do that. The second part that we're trying to get at this we came from completely random question of trying to characterize sugar metabolism in the bladder with collaboration with Lisa. And we came across some enzymatic activities, which seems to be intrinsic in many urinary commensal lactobacilli, which seems to be digesting some of the linkages and sugars which are present in estrogen modified molecules, which are known to be circulating because estrogen has different set of modifications, right? And they also sometimes have sugar attached to it. And it might be that some of the urinary commensals or vaginal commensals, for that matter, may be affecting how much of the active hormonal levels we actually have in particular niche. And I believe it might be very important because we have a just chemical affecting microbial growth and of course affecting also host environment immensely. But we also have a microbe which can affect the host secreted chemicals. And so that we have kind of a circle, which I think we're trying to tease apart. And another project, which we are very lucky to collaborate with Vanderbilt University, with Dr. Haji Francisco, who is studying uropathogen and their invasion in vaginal epithelium. And so we're basically trying to combine acid resistance, interaction with the vaginal microbiome and vaginal host cells, and how that prepares uropathogen to either infect or not infect, how to make it more acid resistant or lactobacilli resistant or less. So ask in a couple of years, I hope we will know more, but it's very exciting because we're trying to get in the aspect not only the hormone, but also how the adjacent niche is changing, how they're changing pathogen behavior as well. I hope I answered some of that question.
Any other thoughts? I have one more question. Oh, go ahead. Somebody. Well, we got a quiet group today.
Weekend. Sorry.
Yeah, I know. So have you thought a bit about the other, a different side of this in terms of potential genetic polymorphisms that may be associated with those women that have recurrent UTI versus those that don't? Especially in light of these particular genes or whatever and their interaction with hosts, the products and their interaction with host responses.
Coming from material sciences, I'm trying not to think about hosts too much, but everything forces
know.
me because for in particular UTIs and uropathogens, it seems to be that if there would be any single binary or like single action or parameter, right, we would already figure that out. And it does seem to be that the combination of effects should be coming. Yes. And it seems to be what we're trying to add, the complexity of microbiome. And what was already initially there is the diversity of host and diversity of uropathogen. So maybe combining these three aspects, we finally will find what the combination which leads. So I don't think that urinary microbiome would be the sole definer because for obvious reason, if you have different hosts, they are colonized by different microbes for a reason, either by the immune response or property of the epithelium, right? Or lifestyle, whatnot. And once it overlays with a difference of pathogen or particular condition or immune state at this moment, that would be interacting. We only, at the moment, approach the question from the uropathogen part. For example, we're taking particular factor from the plasmid encoded, which seems to be serum resistance factor. And we're trying to see how that may be affecting urinary tract infection going to the kidney and sepsis. That's serum resistance would be important for pathogen then.
Sure.
But then we need to move further and add the host finally to that picture. Yes.
Well, let's keep talking because we certainly have patients with these conditions. And as we've talked before, I think we could get some really interesting stuff going together.
And the whole pilot studies will help us to secure funds so we can collect with more site and develop maybe even some tests with the new ideal probiotic candidate.
Exactly.
Thank you so very much. That was I'm sorry I spoke too fast and lost most of the audience, but I hope the main message that it's complex came across. So I'm really excited. Thank you so very much for inviting me.
No, Tanya, thank you so much. I'll be in touch. I'd like you to continue to collaborate with CWRH, and I'll be in touch with you about that.
I promise to learn this letter combination eventually, but and I wish everybody good Memorial Day weekend.
Everybody have a great weekend. Thank you for being on today. Bye-bye.